Millions of insects, each with no plan and no leader, somehow merge into a single living mind, solving problems no individual could. Inside every ant colony, invisible chemical signals pulse like a nervous system, orchestrating traffic, farming, even war, as if evolution rewrote the rules of individuality. What if the neat boundary between organism and colony is a biological illusion? To find out, we have to follow the molecules that turn chaos into intelligence. Inside an ant colony, information moves not by sound or sight, but through invisible chemical trails. These trails are built from a vocabulary of pheromones, each with its own volatility, persistence, and message. Some, like alarm pheromones, vanish in minutes, diffusing quickly through the air and triggering instant action. Others, such as foraging trails, linger for hours on damp soil or leaf litter, their molecules slowly fading, marking the way for hundreds of returning workers. Macro-cinematography, slow tracking shots over the forest floor. Orchestral soundscapes swell, underscoring the invisible logic of the colony. A single colony can deploy more than a dozen distinct pheromone blends at once. Trail pheromones, often made up of 5 to 20 compounds, encode not just direction, but freshness and quality of resources. Alarm signals, by contrast, are simple and fleeting, built for urgency, not memory. Queen pheromones, complex and persistent, maintain social order by suppressing reproduction in workers. Ants detect these signals at astonishingly low concentrations, down to 1 part per billion. Specialized sensory cells on their antennae can pick up gradients as fine as a few molecules per cubic centimeter. The exact blend and strength of a pheromone, along with how fast it diffuses or decays, determines whether an ant will follow a trail, recruit others, or launch an attack. These chemical channels act as the colony's nervous system, encoding time and space without a single neuron. Information travels as gradients, fading and shifting, yet always guiding the collective. In this world, a molecule's lifespan and its journey across a forest floor can shape the destiny of millions. In 1959, the French entomologist Pierre-Paul Grasse introduced a new word to biology, Stigmergy. The idea was as simple as it was radical. In ant colonies, coordination does not require a leader or a master plan. Instead, each ant leaves traces, chemical marks on the ground, tiny changes in the environment. These traces in turn guide the next ant's behavior. It is a feedback loop in which the environment itself becomes the message board, storing and relaying instructions without conscious intent. Deborah Gordon's fieldwork in the Arizona desert brought this principle to life. She watched harvester ants navigate crowded trails, where bottlenecks could choke the colony's food supply. There was no traffic cop. Instead, as the density of ants increased at a junction, each ant responded to the local crowding by laying down more pheromone or slowing its pace. This simple rule, to match your pace to the density, scaled up to regulate the entire colony's foraging flow. When traffic threatened to jam, ants instinctively throttled back, preventing chaos and keeping resources moving. Stigmergy explains how positive feedback can amplify a single ant's action into a collective wave. A faint trail, once discovered, becomes a highway as more ants reinforce it. If food disappears, the trail fades and the traffic dissolves. The logic is algorithmic, rooted in chemistry rather than cognition. Each ant follows local rules, but the sum of their actions produces a living algorithm, one that flexes and adapts, balancing efficiency with resilience. Grasse and Gordon's insights reveal a hidden order. The colony's intelligence is not in any one ant, but in the flow of information through their shared world. Macro cinematography captures a river of tiny bodies moving through the leaf litter, orchestral soundscapes rising as we descend. Deep in the soil, a leafcutter ant colony pulses with hidden purpose. Workers stream back from the forest, each carrying a fragment of leaf far larger than themselves. These leaves are not food for the ants. Instead, each scrap is delivered to a subterranean chamber where a living garden waits. Here, the true heart of the colony is not an ant at all, but a fungus, Leucoagaricus gongylophorus. This fungus is cultivated with the care of a vital organ. Workers chew the leaves into a mulch, inoculate it with fungal threads, and meticulously weed out contaminants. The fungus digests the plant matter, breaking down tough cellulose into sugars and proteins. In return, it produces swollen hyphal tips called gongylidia, packed with nutrients which the ants harvest and feed to their larvae. Macro cinematography lingers on the tunnels, orchestral soundscapes softening. This partnership forms a closed metabolic loop. Waste from the garden is removed, sorted, and sometimes recycled as fertilizer for new fungal beds. Even the youngest members play a part. Recent discoveries reveal that ant pupae secrete a milky fluid during metamorphosis that is rich in sugars, amino acids, and antimicrobial compounds. Adults and larvae consume this fluid, linking development and nutrition across generations. If left uncollected, the fluid fosters mold and threatens the colony's health. The result is a system where every stage and caste has a role in sustaining the whole. Visual cue, macro cinematography on the garden, orchestral soundscapes swell. Within these chambers, the boundaries between ant and fungus blur. The colony's survival depends on the health of its internal garden, just as a body relies on its organs. Nutrients flow from plant to fungus to ant and back again, a seamless circuit of energy and matter. This is not just cooperation, but integration, a living factory where chemistry becomes physiology, and the colony itself becomes a single, breathing superorganism. For those who study the genetics of cooperation, the incentives are made plain by Hamilton's rule, which states RB is greater than C. Here, R stands for genetic relatedness, B is the benefit to the recipient, and C is the cost to the altruist. In the world of leafcutters, high relatedness among colony members aligns individual interests with the health of the collective, making extreme forms of investment in the garden and in one another evolutionarily stable. Macro cinematography pulls back to reveal the nest as a network, orchestral soundscapes resolving into a single note. The fungus factory is a testament to biological emergence. It forces us to ask where the individual ends and the collective begins. When organs, microbes, behaviors, and genes are woven together so tightly, the boundary fades. The colony is neither only ant nor only fungus. It is an embodied feat of integration, a living economy in which identity and function are distributed, and intelligence is found in the pattern of interactions rather than in any single brain. A column of army ants hits a break in the forest floor. Instantly, the front ranks grip the edges and lock together. More ants join, weaving a living bridge that flexes with the colony's needs. As traffic swells, the bridge stretches, sometimes doubling in length as flow jumps from 50 to 200 ants per minute. When the rush fades, the bridge contracts, freeing ants back into the moving mass. Each ant responds only to the pressure and crowding at its spot, joining or leaving as needed. No single ant directs the process, yet the colony's crossing time nearly halves. Fieldwork shows these bridges reconfigure in less than 30 seconds after traffic shifts, mirroring predictions from network theory. Macro cinematography captures the weave of bodies, and orchestral soundscapes swell with each movement. This is collective morphology, architecture built from bodies shaped by simple local rules. The bridge is not a fixed structure, but a dynamic solution, appearing, adapting, dissolving as needed. Under a rotting log, temnothorax ants face a different test. When their nest is destroyed, scouts scatter to search for new shelter. Each scout judges a site, then recruits others, but the colony does not move until enough ants converge. Consensus forms through encounter rates, and if a scout meets nestmates at a site 10 to 30 times within a few minutes, she shifts from slow recruitment to rapid transport, carrying nestmates and brood. The threshold flexes with colony size and urgency. Macro cinematography follows a single scout in close-up, and orchestral soundscapes build as decisions escalate. Thomas Seeley's research with honeybees uncovers a similar pattern. In a swarm, bees debate nest sites through dances. Only when a quorum of scouts, just a few dozen among thousands, gathers at one spot does the swarm commit. In every case, the group decides without a leader, balancing speed and accuracy through distributed intelligence. When genetics enter the conversation, biologists invoke Hamilton's rule, stated as RB is greater than C, where R represents genetic relatedness, B is the benefit to the recipient, and C is the cost to the altruist. Macro cinematography pulls back to show the swarm as a single moving organism, and orchestral soundscapes resolve into a quiet, reflective tone. Biologists have long debated what makes something an individual. E.O. Wilson and Bert Haldebler proposed a checklist: integrated metabolism, coordinated reproduction, and division of labor. Ant colonies pass each test. Their metabolism scales sublinearly with size. Energy use rises, but less than you would expect if each ant acted alone. In colonies from 100 to 50,000 workers, the per-ant energy cost drops by up to 30%. This pattern mirrors Kleiber's law for animals, hinting at a deeper unity. Hamilton's rule gives the math for altruism. Hamilton's rule says R times B is greater than C. R represents the genetic relatedness, B the benefit to the recipient, and C the cost to the altruist. In ants, sisters share about 75% of their genes, so R is 0.75, and helping the colony pays off in evolutionary terms. Workers forgo their own offspring, investing in the queen's brood because the genetic benefit outweighs the cost. The colony's boundaries are drawn not by skin, but by flows of energy and information. When the numbers are run, the verdict is clear: the superorganism is a biological individual. At the moment, the graphic of R times B is greater than C appears. Macro cinematography and orchestral soundscapes converge, underscoring the colony's invisible intelligence. Colonies are not the only collectives that blur the boundaries of individuality. In the soil, Myxococcus xanthus bacteria swarm together, forming fruiting bodies that rise from the surface like miniature cities. Each cell contributes to the structure, some sacrificing themselves so others can survive as spores. Slime molds, for example Dictyostelium discoideum, spend most of their lives as solitary amoeba. When food vanishes, thousands merge into a single slug, guided by pulses of cyclic adenosine monophosphate. This slug crawls as one, then transforms into a stalk and a spore cap, cells trading their independence for a fleeting multicellular existence. In the ocean, the Portuguese man-o-war drifts with the currents. It looks like a single animal, but is actually a colony of zooids, each specialized for feeding, defense, or reproduction, fused into a body that cannot survive apart. Macro cinematography reveals sweeping patterns, and an orchestral soundscape swells beneath the images. These examples stretch the concept of individuality. Where does the organism end and the collective begin? The answers are as varied as life itself. Right now, scientists are decoding how collective intelligence outperforms its parts, reshaping robotics, AI, and even medicine. Macro cinematography frames the choreography of ants, and orchestral soundscapes swell as the colony moves as one. The boundary between individual and superorganism blurs further each year. What we learn from ants may soon redefine what it means to think, to act, and to be alive. Share your perspective below.